Hello student, welcome to EPG Pathshala. I'm Dr. Ashish, Scientist E from Institute of Nanoscience and Technology, Mohali. Today we are going to discuss about the module that is design of nanostructure from nucleic acids under the paper Nanobiotechnology. So after completing this module, you should be able to understand the chemical toolbox to process these different DNA or nucleic acids, different enzymes, how you can actually cut, paste, like this is the required, because if you want to build a structure, a nanostructure, it is very important to know what are the tool you have. So we would know about different enzyme like restriction enzyme for cutting the DNA, or if you want to ligate two different fragments, we would know what are the ligase. Then if you want to copy, cut, copy, paste, so it's simple like that. Restriction enzyme, ligase or DNA polymerase. So this we'll get to know. That is the first thing. So we know about chemical toolbox of doing this DNA processing. And then we would actually, now once we know that, second thing we'll learn is that what are the different strategy to design this kind of DNA or RNA nanostructures with, they will have a sticky end or blunt end, so that the sticky end they can actually stick together with another fragments with a sticky end, and it can form different structure. It can form so normally DNA is one dimensional, right? So if you can actually assemble DNA such a way that you can actually get two-dimensional lattice in 2 dimension, or you can actually make something a cube, or let's say a ball, or a different kind of, if you look at this, we'll also look at towards the end DNA origami, like Japanese, they make very nice 3-dimensional structure with folding paper. So like that, can we do with DNA? So these are different approach or the strategy to make DNA nanostructures. We will get to know about them, and towards the end, similarly, we will also look at 1 or 2 examples of RNA nanostructures. So first, let us understand what is DNA nanotechnology. So far, we all know from our biochemistry knowledge or organic chemistry knowledge that this DNA, they can a particular segment of DNA, they can actually code a particular amino acid. Okay, and there are something called codons, stop codons. So DNA has been looked by us, it's like a genetic material. But now what we are going to look at is that since DNA, we have seen in earlier module that DNA is one particular DNA of let's say 20-30 base pairs. They would be having a length of around, let's say 2 nanometer or so. And so what we want to do is now using different DNA fragment, we want to do self-assembly, and we want to go in a bottom-up way, so that short nucleotides can be organized into a much bigger nanostructures, and which are stable structure. And in DNA nanotechnology, this is one good thing about it is that because of this base pairing, and base pairing guanine can only go with cytosine, right? And adenine can go only with thymine. So it's very specific base pairing. So then sequence of bases is very important when you talk about this another complementary sequence, because that completely determines the structure formation. So we can get very specific desired conformation, and because of this this matching, the correct matching base pair is energetically favorable. Otherwise, if there is a mismatch, then that is not energetically favorable. So then what happens, that nucleic acid that binds to each other in one particular conformation, which maximizes the number of matched pair, because it minimizes the number of mismatched pair in other way. Right. So this kind of DNA nanostructures, this has huge applications, and the field is still developing. A lot of new applications are coming, because one can actually make DNA directed or RNA directed assembly of nanomaterial, and where DNA can act as a template. Okay, and DNA can also act as a scaffold for chemical reactions or single molecule analysis. Okay, another thing is that this different DNA nanostructure, they can also act as a nano container for drug delivery, because DNA is many of the DNA will be biocompatible. And then this can be also used as an antimicrobial devices. You can also use it in the NMR imaging. You can use DNA for biosensing of sensing, let's say, oligonucleotide or complementary sequences. You can also use DNA for computing, so, and this is what is actually you are going towards, which Richard Feynman actually predicted long time ago, that DNA can also be used as an storage material. Let's understand the chemical toolbox to process DNA. Now there can be different enzymes which are essentially protein. There can be different enzymes like restriction enzyme, ligases, helicases. So you can see here it's a double-stranded DNA, and if you have to cut this DNA double strand at a particular sequence, you need restriction enzyme. So restriction enzyme basically cuts DNA at a specific sequence. Now you can see the other structure where it talks about ligation of sticky unit. So if you have two complementary sequence which are sticking out from the cut DNA double strand, and you want to join them together, so you need some enzyme called ligases. Then ligase actually binds to that particular enzyme that binds to that particular segment of DNA, and it repairs. Like that, there can be a helicase enzyme which actually unwinds the DNA, or there can be also, we will also look at later what is the role of DNA and or RNA polymerase. How do they make copies when we talk about polymerase chain reactions? Let's look at what is restriction enzyme. So restriction enzyme basically cleaves DNA into fragments at or near a specific recognition site within the molecule. That particular recognition site is known as restriction sites, where the restriction enzyme binds. Normally these recognition sites, they have something called palindromic recognition sequence, so which is nothing but it reads the same in the reverse strand and as it does in the forward strand. So if you look at the following example, where you look from 5' to 3' end of a oligonucleotide, you have GAA-TTC, but if you look now the double-stranded DNA, if you look the other strand which is in other opposite direction, you will see it is actually running the same sequence. So that's what restriction enzyme that can actually read, and it binds to that particular segment, and once it binds to that particular segment, it can actually you can see there is a restriction enzyme that is called EcoR1 that actually cuts just after that guanine moiety, guanine and adenine moiety, that particular phosphate bond there, that actually breaks. And you can see from different chart and also from the cartoon that there is also a sticky end. Now this sticky end, when they cut, the sticky end which comes out, protrude out from the DNA double strand, it has complementary sequence. Now you can look at in the table, there are different restriction enzymes, and they actually recognize different recognition sites, and they also cut at different phosphate sugar bond. So when we are talking about joining DNA, then we need this enzyme called ligase enzyme. So what it does is, you can see that there are two DNA strands. One has this 3' end, the phosphate group, 3' end you have the hydroxyl group of the sugar, and which should actually join with the DNA strand at the 5' end of which we have the phosphate bond. So basically you have to make a phosphodiester bond, and this is where DNA ligase actually binds, and it actually repairs and form the bond, this phosphodiester bond. So if you look at this example, when you have a single stranded DNA, it can actually repair the single stranded DNA, and the double stranded DNA, it you can see those cut section, it repairs to form, the ligase repairs to form perfect double stranded DNA. Let's look at polymerase chain reaction, which is very famous, and this is where DNA or RNA polymerase, that particular enzyme, is useful to make copy of DNA. This is, sometimes it is important that if you want to increase the sensitivity of the system, you need to have, instead of one particular DNA, you need to have thousands of DNA. So that actually increases the sensitivity of the system. So that's why you need to replicate that particular DNA, thousands of millions of copies of that particular DNA. So if you want to do this polymerase chain reaction, you need to have, along with the particular DNA you want to copy, you need that double-stranded DNA. You need also all the mononucleotides. You need the DNA polymerase enzyme, and you need something called primer. Now the primer would actually have certain segments of the DNA strand as complementary, only certain segment, not the entire one. Now what it does, now this entire polymerase chain reaction that has 3 different steps, and these steps gets repeated again and again. First step is the denaturation step. When you take this double-stranded DNA, you heat it up to 90, more than 90 degrees, then DNA double helix get denatured because of disruption in the hydrogen bonding, and it forms single-stranded DNA. And in the reaction mixture, you have now primer. You can see the green or the blue primer, they bind to that segment of the DNA wherever it finds complementary, and then you basically anneal. So when you anneal the system, heat and cool at 50 degree, you keep it there, so it forms this DNA double helix at that particular segment, and then DNA pol actually binds to that particular area, and it actually copies. It's like a Xerox machine. It actually keeps taking the mononucleotide and it keeps forming the complementary strand, and then that goes on in 72 degree, and eventually you make also complementary strand of each of the DNA strand, with the mother DNA strand. And after that, again you repeat the same procedure. That means again denature this particular double helix, and then again you go through annealing and elongation, and you get to form. So you can see after first cycle you can get 2 of the daughter DNA strand. After second cycle you get 4. Like that, if you repeating the cycle again and again, you actually can get 2 to the power n DNA fragments, where n is the number of cycle you repeat. So that actually exponentially grows. So that is very special of the DNA polymerase reaction. Now when we talk about structural DNA nanotechnology, it actually consists of combining these unusual DNA motifs with sticky end, that we can actually cohesive hydrogen bonding interaction, that the sticky ends can actually join together and it can form DNA double strand. And you can see this example by Professor Siemens group. They showed that they form again the bDNA with the sticky end, and you can see the red part that's in the crystal structure of the double-stranded DNA. The red part, it shows joint part of the double-stranded DNA sticky end. So far we have discussed one-dimensional DNA formed by the sticky end. But now if we need to know about this branched DNA junction. Now in the branched DNA junction, you can see this is a stable branched DNA junction which can actually be formed by 4 strands, and if you look at some segment of strand 1 and strand 2 is actually complementary, whereas the other part of the strand 2 that is complementary to DNA strand 3, and they can actually form this kind of branched DNA segment. And these junctions, they are also known as Holliday junctions. And you can see the structure of the Holliday junction, they actually look like the structure of this RNA secondary structure because of this intramolecular loop and stem formation. And on the right hand side you can see actual DNA double helix of these 4 different color coded DNA, and you can see clearly the DNA double helix, how it actually forms this Holliday junction. This is a classical example of Holliday junction formed by 4 different strands of DNA, where 2 of the strands are forming already DNA double helix. Now when we talk about this DNA branched junction, if this branch junction, they have all of the branch segment, they have their complementary strand. You can see from here it can be X, X dash, Y dash, Y. These are all the sticky end coming out of the DNA branch junction. Now they can actually form 2-dimensional DNA nanostructure, and it can actually form eventually 2-dimensional helix because of this cohesion of these sticky ends. And you can see in the bottom there is an example which was published in Science. It shows this formation of 2D DNA nanostructure using this kind of Holliday junction with sticky ended complementary sequence. And you can see from AFM lower magnification a very nice fiber-like material. However, when you zoom this fiber-like material, you can actually see two-dimensional lattice, like net-like lattice formation because of this branched sticky assembly. So we have looked at how to assemble DNA nanostructure into two-dimensional lattice. Now let's make the system even more complicated. We can actually assemble this DNA nanostructure in 3 dimension. So how to do that? So you see, this is a very nice approach where you take 3 different types of DNA single strand, and they can actually stepwise assemble. Now one of them can already form this branched junction, and the combination of all the 3 DNA strand, they can actually form 3-point star motifs. So that can be 2 different types of 3-point star motifs you can see over there. And now this 3-point star motif, they are acting as a primary building block, and this building block at different concentration can give rise to different DNA polyhedra. So if 4 of these molecules they come together, and they can form a tetrahedron structure, and that can only be possible when you take this DNA strand at around 75 nanomolar concentration. Much lower concentration you can form tetrahedron, but 20 such 3-point star tiles, when they come together, then you can see you can form something like dodecahedron. But see, the concentration regime is different, but at very high concentration you can see you can form this buckyball like structure. You can see the 3-point star motif is little different for this one, and you need to have around 63-point star motif to form this kind of buckyball structure. So this is unique, and this is concentration dependent. Now formation of these different DNA polyhedra from these 3-point star tiles, one can clearly visualize by AFM or TEM. You can see here the AFM images, and you can see this very clearly. The tetrahedron can be visible, or other dodecahedra or buckyball is visible. You can see the size of the different polyhedras are very different, where tetrahedra is much smaller, 10 nanometer, the dodecahedra 24 nanometer, and buckyball is something like 42 nanometer. And in fact, they looked into the cryo-TEM they recorded cryo-TEM at different angle, and then from there they did a single particle 3D reconstruction using computation. They can actually build up the model, and the dimension of those model that actually matches pretty well with the cryo-TEM or the AFM images, and that was also further verified by DLS, another microscopic technique. We can see that DNA can be really programmed to assembled into well-defined regular polyhedra, which can be used as synthetic nanocontainer or 3-dimensional structural scaffold. We already know about the term origami, which is a famous Japanese term, and which depicts arts in 3 dimension. So we can similarly, just like origami, and that is possible because of this presence of complementary base pairs, and that makes DNA a very useful construction material. Now, DNA origami is a bit more complex process, and if you want to perform your DNA origami, and the design involves normally 5 steps, where the first 2 steps you need to sit and do it by hand, and then last 3 steps you need to be aided by the computer modeling. So let's look at the first 3 steps. What we do is we first need to build a geometric model of the DNA structure that will approximate the desired shape, and you can see the cylinders. Now each cylinders they represent the parallel DNA double helix, and these different cylinders, they are joined together by something crossover region, and eventually in the second example you can see that it has scaffold crossover. Using this crossover you can have these crossovers where actually the DNA twist can take place, and eventually in the third place what you can do, you can actually have the staple strand, which you actually staple it, and in the staple strand actually provides the Watson-Crick complementary hydrogen bonding for the scaffold creation. So thus, if you look at this DNA origami shape, if you want to idealize these three different structures, so one is the first one is a square-like structure, or a star-like structure, or a disc with three holes in it. So first of all, you have to make this geometrical model, which is and then you have to define this crossover region, which is given by these different color code, and then you can see the crossover regions, and eventually you can actually fold the DNA accordingly to get this kind of AFM image, and you can see the AFM image nicely matches with the origami shape which intended to do. So we have seen different nanostructure formation in 2-dimension and 3-dimension from DNA. Similarly, RNA can also form RNA nanostructures, and one famous example of RNA nanostructure is something called Tectosquares, which is also found in tRNA. And this also similarly, it can have this intra-strand secondary structure formation, and you can see that that can be this A, B, C, D different secondary structures, and these secondary structures can actually, in presence of magnesium, it can form this something like this Tectosquare. C shows the 3-dimensional construction of the Tectosquares, and you can see Tectosquares also have different 4 sticky end, which actually can also join. Ideally, it can form 2-dimensional lattice structure. Here you can see formation of these Tectosquares, which can be utilized into RNA nanostructures in different dimension. In one of the example here is that this nanostructure can be assembled in ladder form. You can see it on the left, or it can also have net-like structure where it can actually form 2-dimensional lattice. This, if you look at the bottom, the images, that they're actually AFM images. The bottom ones you can see is high magnification AFM image, where you can see both ladder-like structure, and in the right case you can see the net-like structure of 2-dimension lattice with repeated periodicity gap between the Tectosquare. So if we look at the possibility of this DNA nanostructure, what we have seen for drug delivery, we see there are some important issues. First of all, this when you talk about this DNA, you have this negatively charged phosphate backbone, and what happens is that if you have to permeate through the cell membrane. Now cell membrane is also having this phosphatidylcholine which are also, so they can repel. So in order for you that you want to do drug delivery or delivery through the cells, then what you have to do, you have to compensate this negative charge of DNA by adding some kind of transfection agent, which actually can eventually take the nanostructure, get inside. Now there are some superior feature of DNA nanostructure. First of all, because of its specificity and forming this complementary base sequence, you can actually have very predictable and very well defined structures, and they would be of uniform size and shape. That's number one. Number two is that this DNA nanostructures, they can actually have very high load of high capacity of cargo loading, and this nanostructure can be very easily internalized by the cell, and in physiological condition this DNA are stable. That's most important. And this again, I am mentioning this DNA nanostructures, they have excellent biocompatibility. So let's look at the application of this DNA nanostructure in drug delivery. So here this M13MP18, this is a DNA long strand, and that has been, with the use of annealing with staple strand, can actually form 2 different types of DNA origami structure. It can form either tubular structure, or it can form 2-dimensional triangular structure. Now, so these DNA origami structures can be loaded with doxorubicin. Now doxorubicin can actually intercalate with the DNA bases, and it can taken up by these structures. And now this doxorubicin, you can see more or less the tubular structure of DNA origami or triangular DNA origami structure to AFM image. You can see that they stay intact. However, when they were taken on tumor cells, it was shown that a very high level of drug loading efficiency was first achieved, and there is a prominent cytotoxicity, human breast adenocarcinoma cancer cells, this kind of doxorubicin loaded DNA origami structures. So definitely DNA origami enhances the cellular internalization of the doxorubicin. So if we summarize the timeline and progress of the DNA nanotechnology, it's all started with 1980s when this DNA double strand with the Holliday Junction, scientists started to use the sticky end concept to form two-dimensional or one-dimensional lattice. Then came these three-dimensional DNA cube or polyhedra, different kind of three-dimensional structure. Then came DNA origami, and then there associated with it a lot of applications. But so there has been a lot of efforts on design initially, but eventually when it came in the recent time, it's all about the aesthetics and the application driven design which got the priority. So let's summarize what we have learned so far from this module. So first of all, we have looked in detail the different cut, copy, paste tools in order to process DNA, different enzymes, and if you have to, then we learned about palindromic sequence, how do restriction enzyme they read that and eventually cut, looked at different restriction enzymes. So we know if we have to cut at a particular position, then which particular restriction enzyme you should take. We also looked at ligase enzyme and how ligase enzymes can be used to really heal the DNA strand. PCR is very important. We also looked at in that aspect DNA polymerase, how it can copy. These are important things because if you have to multiply the DNA nanostructure and you have to get a lot of it, then you need to really do PCR. Now, having done that, having understood the processing tools, then we started cutting those DNA double helix into a specific sequence so that we can have something called sticky end, and this sticky end then can be coupled to different DNA helix with the sticky end. They can come together and form nanostructure. Now then we also looked at this branched DNA double helix, then that can actually form these Holliday junctions, and these Holliday junctions eventually with a sticky end can form this two-dimensional helix. Then we looked at more and we also looked at these 3-point star tiles or motif, so that we can actually fold the entire DNA these nanostructures or into three-dimensional polyhedra. It can be cube, it can be tetrahedron. So this is the strategy we have looked at. Lastly, we also looked at a very complicated three-dimensional strategy for complex designing of DNA origami, and what are the approach, how to go about it, some basic idea we got about DNA origami. And lastly, using the same similar idea just like in DNA, we also looked at the nanostructure designing from RNA, and we towards the end we looked at some example of these DNA origami based structures as nano container for drug delivery and how that can be beneficial. So I hope this will be useful for your knowledge. Thank you.